regions. So here's antibody. The V region contains the binding site, which binds and latches this particular pathogen, because this particular pathogen has triangular looking antigens which are complementary to the antibody's variable region, the antibody's binding site. And so as you can see, the antibodies are highly specific and so binds to a specific type of antigen. So let's look at this connection, the contact between antibody and antigen in a bigger picture. So here we have a pathogen with its specific type of antigen on it. And here we have the antibody. The antibody consisting of a light chain and heavy chain. The top portions are the variable regions and the rest are the constant regions. So here's antibody, and the V region contains the binding site, which binds and latches, makes contact with the antigen of a pathogen. So let's have a closer look at this heavy chain of the antibody binding to the antigen. So here we have the structure, the protein structure of the antigen of the pathogen, and here we have the heavy chain variable region of the antibody. And the site of which the antigen binds or makes contact with the heavy chain binding site is known as the epitope. So the epitope binds or makes contact with the binding site of the variable region, essentially. And that concludes the video on the diversity of antibodies and the T-cell receptors. I hope you enjoyed that. But if you click on the links provided, and I know I said that in my last video, and ask questions, answer questions, and post some interesting things such as artworks. Please. And you can also change the quality settings to the highest one, such as 720, I guess, for better graphics. And in this video, we're going to talk about antibody somatic VDJ or VDJ recombination. And this is going to be part 1. And so antibody somatic recombination occurs in B cells. And going back to the B cell development, we begin with a progenitor lymphoid cell, which through somatic recombination, also known as VDJ recombination, will make an immature B cell with a unique antibody that binds to a specific antigen. But this in itself is a very simple diagram. It's actually a bit more complicated in that first, the progenitor lymphoid cell can become a progenitor T cell, or in this case, what we're actually looking at is that the progenitor lymphoid cell can become a progenitor B cell. And once it becomes a progenitor B cell, it will go through somatic recombination of the heavy chain. So there's heavy chain rearrangement. Typically, the progenitor B cell will make an immunoglobulin M antibody. And so this progenitor B cell will become a precursor B cell after somatic recombination of the heavy chain. When it becomes a precursor B cell, the precursor B cell will then undergo another somatic recombination, but this time of the light chain, so light chain rearrangement, to become a immature B cell. So as you can see, there are 2 somatic recombinations occurring. 1 in the heavy chain and 2 in the light chain. So let's go back to where the progenitor B cell will undergo its first somatic recombination. So here we have the DNA of the germline, so germline DNA of the progenitor cell. And in this germline DNA of different types of chromosomes, we have some genes for the heavy chain. So remember, somatic recombination first involves the heavy chain. And in the heavy chain gene will essentially make the heavy chain of the antibody, if you don't remember. And this is the heavy chain of the antibody. Now going back to the heavy chain gene, we will look at the overall picture of the VDJ recombination, which involves the VDJ segments. In reality, there are many V segments, many D segments, many J segments, but we'll only look at one to understand the process here. So in the heavy chain gene, we have a leader segment, we have a V segment known as the variable region. so the variable region. And we have the D segment, the diversity segment. We have the J segment, the joining segment. And we have the constant region. On the heavy chain gene, the constant region is known as constant mu, which will essentially make the antibody for immunoglobulin M. And now this is in the DNA. And the DNA contains introns, obviously. So a point to make is that the J and C region are in close proximity. However, the variable region and the diversity region are far away from each other. So the first process that occurs is that we have J and D recombination in the heavy chain. J and D recombination is where the J will bind to the D region, or the D segment, to bring C in close proximity. Next, we have VDJ recombination, where the D and the G segment will bind to the variable region, so bringing the C, the constant mu, in close proximity with everything else. Now the constant mu, here I have drawn a bit bigger, because a constant mu actually consists of many segments. For immunoglobulin M, there are 4 constant regions, because the heavy chain consists of 4 constant regions. Following VDJ recombination, this will actually produce transcribe for an RNA. So this whole sequence now of VDJ and constant is an RNA. And because it's an RNA, we still have introns, and from splicing, this will remove the introns within that particular gene. And then once the introns are removed, it'll become mRNA, which will then make protein. And now this is essentially the protein for the heavy chain of the antibody. Unfortunately, the D and J regions, I'm drawing here in orange and green, are drawn incorrectly. They're supposed to be on the other side, on the opposite side. But that's just a minor error. Anyway, this antibody is essentially this IgM antibody we find in this immature B cell. But how about the light chains? Because we don't have the light chains yet. Well, after the heavy chain recombination has occurred, we get a precursor B cell. And in the precursor B cell, we have a gene for the light chain. And so the light chain gene can undergo somatic recombination, VJ recombination. So in the precursor B cell, we have the light chain gene. Now the light chain gene is different to the heavy chain gene in that the light chain gene does not contain a D region. So we still have the variable segment, the joining region segment, and the constant segment. But we don't have the diversity segments. We of course have introns in this gene because it's the DNA. And remember, it's still the same. The joining region and the constant region are in close proximity, but the variable region is far away. Now what first happens is we have the J and V recombination, where the joining region and the variable region bind together to bring the constant region in close proximity. And essentially after this it gets transcribed to make RNA, which we have here, with a poly A tail. Now this RNA with the V and J bound together and the C in close proximity will then undergo splicing, the removal of the introns, simple enough. And so now we are left with an mRNA with the leader V, J, and C regions, which will essentially make the protein. And so as we've seen, the light chain gene has made a light chain protein, which will make this particular antibody. So the light chain fits this antibody, V and C regions, with the joining region here. Now, as I've mentioned, we don't only have one variable region, even in the light chain, one joining region, constant region. There is actually many V regions, many joining regions, many diversity regions. And so all this mixing up between the V, D, and J will make a unique antibody. Not only that, during somatic recombination, new nucleotides can also be added to increase the diversity and specificity of these antibodies. So I hope you understood this first part of the video. Second part, we'll look more deeper into somatic recombination, VDJ recombination. Thank you. Under Hasurugan Biology and Medicine videos, please make sure to subscribe, join the forum and group. For the latest videos, please visit Facebook Armando Hasurugan and like. And also you can ask questions, answer some questions, and post some interesting things, including artworks, please. And change the quality settings to the highest one, such as 720p for better graphics. We're continuing on somatic recombination, also known as VDJ recombination, for antibodies. So as we've learned, progenitor B cells makes immature B cells. And a process called somatic recombination, or VDJ recombination, provides a unique antibody for the immature B cell. So for example, a progenitor B cell makes an immature B cell, and another progenitor B cell makes another immature B cell. Important information to know is that there are two types of light chains. Not two types as in the genes shuffling within the light chain gene, but there are actually two classes of light chains, as in two classes. So for example, if we look at these two immature B cells and we compare the antibodies, they both have antibodies. However, the light class is different. The light chain type is different. One is lambda light chain and the other one is kappa light chain. And so as you can see, this would increase the diversity of these antibodies. So if there's two light chains, two types of light chains, this would mean that there are two types of light chain genes. However, there's only one type of light heavy chain, which consists of many type of constant regions. But we won't get into that right now. So, for example, if we go into this progenitor B cell, chromosome 22 has the lambda light chain gene. Chromosome 2 has the kappa light chain gene. And chromosome 14 has a heavy chain gene. So as you can see, with the two types of light chain and the heavy chain mixing up, we can have a diverse types of antibodies. So for example, if we look at the chromosome 2 kappa light chain gene and the chromosome 14 heavy chain gene, we can see how they can mix up to produce a unique antibody. So let's begin with the heavy chain gene. As I mentioned in my previous video, there's not only one variable region, one diversity region, one joining region. There are actually many types of variable, diversity, and joining segments. And so for example, in the heavy chain gene of chromosome 14, we have about 1 to 40 variable regions, 1 to 23 diversity segments, and 1 to 6 joining segments. And we have the constant mu, which will essentially transcribe for immunoglobulin N antibody. So as you can see, the mixing up of these variable regions, diversity regions, and joining segments will create a unique antibody. So what first happens with the heavy chain gene is it goes through DJ recombination, which essentially is where one of the diversity segments and one of the joining segments bind together. So in this case, we have here diversity segment and joining segment binding together. Next, what happens after DJ recombination is VDJ recombination, where essentially the previously bound D and J will bind to one of the variable segments and then be transcribed. So in this to a V1 variable segment 1, and then we have the diversity 2 and joining 2, and the constant mu actually consists of 4 constant regions. And essentially, this is already an RNA. And because it's an RNA, it has to go through splicing to remove the introns, which will make an mRNA, and then a protein. And this protein is the heavy chain part of the immunoglobulin. It consists of variable 1, D2, J2, and the 4 constant regions, which will fit to this structure, which is an immunoglobulin M antibody. So what about the light chain? As we mentioned, the light chain, we're looking at the kappa light chain gene. And both the light chain, the lambda and the kappa, do not have a diversity segments. So the kappa light chain gene consists of about 1 to 38 variable segments, about 1 to 5 joining segments, and a kappa constant region. So it doesn't consist of a diversity segments. And the first process that occurs is JV recombination, where the joining region will bind to one of the variable regions. In this case, a variable segment 2 will bind to joining segment 3, not D3. It's meant to be J3. It's a mistake. And then this is an RNA, and because it's an RNA, it has to go through splicing, the removal of the introns, which will produce mRNA and essentially protein consisting of variable segment 2, joining segment 3, not D3, and a constant kappa, which will essentially make the antibody, the light chain part of this immunoglobulin M antibody. So now, how does the actual recombination process occur? How does the JV recombination process occur, for example, in the kappa light chain gene? So if we have this kappa light chain gene again, consisting of just 1 to 38 variable regions, and then 1 to 5 joining segments, and the kappa constant. So what is the process that actually occurs through this recombination?